In this video, we're going to look at meiosis and its role in sexual reproduction. So remember, as a review, most of your cells have two sets of chromosomes, so we call those 2N or diploid. And that all cells go through the cell cycle where in S phase their DNA is replicated. Again, also remember mitosis, asexual reproduction, makes clones, and makes two daughter cells. Because we're going to compare these two. So, and then finally, mitosis makes micelles. So, growth, repair, replacement. Now, when we look at meiosis, meiosis is also nuclear division, but it's involved in sexual reproduction, meaning two individuals combine their genetic material to create a new unique offspring. So, I like to think of this as meiosis makes me, because we are all the products of sexual reproduction. The cells inside of me, those are made by mitosis, but me, I am made from meiosis. Now, it's used to make what are called gametes. And it's going to be really important because meiosis and sexual reproduction help increase genetic variation. And we're going to see how that works here in a little bit. So what are gametes? Gametes are cells that only have one set of chromosomes. So they are N or haploid cells. And in humans, these are your egg and your sperm cells. So the cells that we use for sexual reproduction. And so again, I like to think meiosis makes gametes. So meiosis makes me, and it makes me through gametes. Now, it works in this way. We have a haploid sperm that has 23 chromosomes, a haploid egg that also has 23 chromosomes, and it's going to form the diploid baby. So when you add together the one set from the dad, one set from the mom, that's how you get the two sets for the baby. So what are the steps to meiosis? Well, this is a little more complicated than mitosis was. We actually have two big steps. So we're going to end up with actually two cell divisions. Now in meiosis I, this is really the important step. This is where it's called the reduction step, where we go from diploid to haploid, where we actually cut the number of chromosomes in half. And it's going to make two cells, going from 1 with 46 to 2 with 23. In meiosis II, we're basically going to do the exact same thing as mitosis, just with haploid cells. And we're going to end up then making 4 gametes. So the end of meiosis has 4 cells, which are all N cells. So in other words, we go from 23 to 23, and we'll see exactly why that works here in a minute. So again, looking at this again, 46 to start. After meiosis I, cuts it in half to 23. Meiosis II simply splits those two and creates 4 haploid gametes. Now let's look at meiosis I a little more closely. What's going to happen is homologous chromosomes are going to line up. And this is a big difference from mitosis. And because of that, I'm going to show you the two processes next to each other. So remember, in mitosis, they lined up vertically so that a sister chromatid could be pulled to one side of each cell. In meiosis, notice how the homologous chromosomes are actually lined up together. So you can see that when they start to separate, only one of each homologous pair is going to go to each side of the cell. So this is how we're going to cut the number in half. So when we look at how this works, when they split, this is how we get that reduction. And then cytokinesis is going to happen, make 2 N cells that have 23 chromosomes. Now the problem is these things still have replicated chromosomes. because they went through S phase and replicated their DNA just like every other cell. Problem is, they didn't really need to. But because we have replicated chromosomes, we got to separate those things. So that's why we go through meiosis II. So in meiosis II, the chromosomes are going to line up, sister chromatids split, nucleus divides, and cytokinesis is going to occur, just like mitosis. It is literally mitosis, but with haploid cells. So again, line up just like mitosis, sister chromatids split, nucleus reforms, cytokinesis, and now I have 4 cells that are all haploid, and these are my gametes. So these would be my egg cells or sperm cells. Notice they have half the number of chromosomes that they started with. So once we make these cells, how does sexual reproduction actually work? And we're talking about at a cellular level, not like in health class. When the 2 gametes, the one from the mom and the one from the dad, come together, that's called fertilization. So they're combining their genetic material, and that creates a new diploid cell, which we call a zygote. So that is the first cell that's going to become a new baby. So after meiosis, we have the haploid egg and haploid sperm. Fertilization makes the diploid zygote. And then mitosis is going to kick in from there because all that zygote is going to be the exact same as all the rest of the cells that make up that baby. So once we make that first cell, mitosis takes over. Now, finally, we need to look at how does meiosis increase genetic variation? And there's 2 processes. We're going to look at crossing over and independent assortment. So crossing over happens in meiosis I, and basically it's where homologous chromosomes exchange or kind of trade DNA. I just think of it like making a trade. So here in meiosis I, when they're lined up, some of the chromosomes are actually going to flip sections of their DNA. This means that we have entirely new chromosomes and entirely new combinations of genes. So this ability to mix some genes up a little bit helps to increase diversity. So here we have variation in our gametes as a result of crossing over. All of them are slightly different. Now, independent assortment simply means that your homologous pairs are going to line up randomly. Your genetics are made from chromosomes from your mom and your dad, and you're going to give your child a mixture of mom and dad. And that's going to be really important, as you're going to see here. So without independent assortment, if they just lined up all the mom chromosomes on one side and all the dad on the other side, you're going to end up giving your kid either the stuff that your dad gave you or the stuff that your mom gave you. And that has major implications, because if that's the way life worked, there would literally only be 4 possible offspring that a couple could make. Now, obviously, that's not how things work, because you could have 20 kids and they all look completely different. So, and that's because with independent assortment, we mix together the mom chromosomes and the dad chromosomes. So now we have a mixture, and then if we add crossing over, every gamete is going to be different. And this is how we increase that genetic diversity. Since every gamete's different, every baby is going to be different. So as a recap, mitosis makes my cells, meiosis makes me. When we look at this in this chart, asexual versus sexual, they both start 2N. Mitosis makes a clone, meiosis cuts it in half. 1 versus 2, 2 versus 4, clones versus gametes. Make sure you really know this chart. Alright, and looking at this in a slightly different way, we start 2N in both. Mitosis is done after 1 division, makes clones, the cells are all the same. Meiosis I cuts them in half, so now we have haploid cells. Meiosis II does it again, and so we end up with 4 gametes.